I would like to start by thanking the organisers for inviting me to present this keynote lecture. I have also taken the liberty of slightly amending the title of my presentation, as I would like to take this opportunity to present my journey of the work that spans a period of over 14 years of how we dissected the cellular and molecular profile of Dupuytren's disease, identified key signaling mechanisms, and how I translated these findings through to phase 2 clinical trials. Dupuytren's disease is incredibly common, but this is the real issue, that treatment options are currently limited to late-stage disease. And we've heard over the last few weeks in this symposium, we can cut it out, we can disrupt it with needles or collagenase. But this is what patients want. They know when they have Dupuytren's disease at an early stage, and currently there's no validated approved treatment. And in our review here in 2016, we assessed all of the available literature and found that in fact there are no clinical trials with control groups and blinding of the participants and the assessors for some of the current treatment options that are used, such as intralesional steroid injection or radiotherapy. What I've just described is rather reminiscent of what we used to see in patients with rheumatoid arthritis back in the 1990s when I was a surgical trainee. Patients used to present with florid synovitis, which we as surgeons used to debulk, and invariably it recurred, and some clinicians suggested that patients should even be treated with radiotherapy. And it was only when this group in 1989 defined TNF as a potential therapeutic target that it totally changed the landscape of how these patients are managed. And they went on to get the Lasker Award, and anti-TNF has essentially now become standard of care, together with a whole host of other monoclonal antibodies targeting interleukin-6 receptor, etc. Today I'm going to describe how my group has dissected out the cellular and molecular profile of Dupuytren's disease and the signaling mechanisms, the crosstalk between the various cell groups, which maintains the disease ecosystem and the chronic low-grade inflammation, how we identified TNF as a potential therapeutic target, the data from a dose-ranging phase 2A clinical trial which has been published, and I'll share with you the top-line data of the phase 2B clinical trial in patients with early-stage Dupuytren's disease. The earliest stage of Dupuytren's disease is represented by these highly cellular nodules, and a single-cell RNA sequencing and cytoph studies show that it comprises mixed population of fibroblasts, myofibroblasts, endothelial cells, immune cells, and pericytes. Let me take you through each of these in turn. First, the fibroblasts. They're not a homogeneous population. There are at least 4 subsets. And I draw your attention in particular to this one, the ICAM1 positive IL-6 high, which are a relatively small population, especially enriched within the nodules, and they play a key role in attracting the immune cells to the site of disease. The myofibroblasts are also not homogeneous. They range from this actor 2 low group through an intermediate to a CD82 high population, which are highly contractile, as shown here in this single-cell contractility assay. And it is these intermediate and CD82 high that are mainly responsible for matrix production and remodeling. The cycling population don't contribute very much to matrix production. Instead, they do exactly what their name suggests. They maintain the pool of myofibroblasts. We also looked at the different sorts of immune cells, and as you can see, there's several different subsets of macrophages, dendritic cells, T cells, and not apparent in this single-cell RNA sequencing study, because the numbers are too small. We also see very low numbers of mast cells, which I'll show you in a moment, are very important. With freshly disaggregated cells from nodules, we see a whole array of cytokines being produced. And I'm not going to take you through all of these, but I draw your attention in particular to TGF-beta, which is the archetypal cytokine associated with fibrosis. You can see about 300 picograms per ml, relatively large amounts of IL-6, quite small amounts of TNF, so around 50 picograms per ml, and even lower amounts of IL-33. And what we then did was tested the effects of many of these in functional assays. And I'm not going to take you through all of those, but let's look at a couple of key ones. I'm going to start by defining the effects of TNF in the various cell populations. So this was particularly interesting because what we found was that at very low levels, so around 100 picograms per ml, in fact the dose response broken down here, so somewhere between 50 and 100, only in Palmer fibroblasts from Dupuytren's patients it makes them much more contractile. But at higher doses it's inhibitory. But interestingly, if you take non-Palmer cells from the same Dupuytren's patients, they don't respond in the same way, and neither do Palmer cells from normal individuals. They become less contractile but at much higher concentrations of TNF. And if you take the myofibroblasts from Dupuytren's patients and expose them to the anti-TNF, so here shown the data for the approved anti-TNF agents that are used for other conditions such as rheumatoid arthritis, you see a downregulation in contractility, which is accompanied by downregulation in alpha-SMA protein and here collagen 1 messenger RNA. The results with TGF-beta are very different. So with this cytokine, it converts all cell types, irrespective of their origin, into much more contractile myofibroblast-like cells, but at much higher concentrations than we see physiologically. So you will recall in those freshly disaggregated nodular cell cultures, it was around 300 picograms per ml, but we begin to see effects now at 1,000 and even 10,000 picograms per ml. So it's acting indiscriminately. And predictably, when you inhibit TGF-beta, you see a downregulation in myofibroblast contractility as well as a downregulation in alpha-SMA protein and collagen 1 expression. I've shown you data that inhibition of either TNF or TGF-beta is effective in downregulating the myofibroblast phenotype. So why select TNF? Well, in freshly disaggregated nodular cells, we see around 50 picograms per ml of TNF and around 300 picograms per ml of TGF-beta in the supernatant. Cells are often subcultured up to passage 5 or 6, but already at passage 2 we see the TNF concentrations falling off because the immune cells have largely disappeared. But through autocrine secretion by the stromal cells, we're now seeing a huge upregulation of TGF-beta production. So this suggests that the TGF levels seen here on later passage cells are really a culture artifact. And this probably goes some way to explaining the failure of targeting TGF in multiple late-phase clinical trials. My colleague Dominic Furness in Oxford and Paul Verker in Groningen have shown the key role of Wnt signaling pathways in Dupuytren's disease through their genome-wide association studies. We went on to show that TNF only in Palmer fibroblasts from Dupuytren's patients acts via the Wnt signaling pathway to upregulate genes such as collagen 1 and alpha smooth muscle actin, typically associated with fibrosis. And so now we have both molecular and genetic data supporting TNF as a potential therapeutic target. We know that the genetics of Dupuytren's disease is incredibly important. There's a heritability of about 80%. But Dupuytren's disease is restricted to the palm of genetically susceptible individuals. So this suggests that epigenetic regulation must play a key role, and indeed it does. So histone acetylation is associated with increased gene expression, and what we have shown is that histone acetylation in myofibroblasts is crucial in maintaining their phenotype. Conversely, histone methylation suppresses gene expression, and our most recent data show that this mechanism is important in regulating chemokine expression by those immune regulatory fibroblasts through an intermediate molecule called PLAG2G2A. So suppression of these genes leads to increased expression of CCL2 and CCL7, which in turn recruits the immune cells, and those produce the TNF that drives myofibroblast phenotype. Dupuytren's disease persists and develops over a period of many decades. Typically, inflammation in humans occurs and resolves over a period of weeks and months. But what we see here in Dupuytren's disease is the inflammation persisting over a very long period. And what we found was that those immune cells produce TNF, which drives the development of myofibroblasts, and these in turn produce low levels of a cytokine called IL-33, which in turn acts on the immune cells, and hence we get this vicious cycle whereby the inflammation persists over a very long period. Our most recent data show that those various cell types that I've described are not randomly distributed through the nodule, but in fact compartmentalized. So you have these immune regulatory fibroblasts, which attract the immune cells, which in turn produce TNF, which drives the myofibroblast phenotype. But these immune regulatory fibroblasts are maintained by platelet-derived growth factor produced by the fibrotic endothelium. And there is this relative segregation of the myofibroblast compartment from the remainder. And we see a very similar pattern here in lung fibrosis in patients with idiopathic pulmonary fibrosis. So segregated myofibroblast foci, and then these immune regulatory fibroblasts and blood vessels around them. Having identified TNF as a potential therapeutic target, I applied to the Wellcome Trust and Department of Health through the Health Innovation Challenge Fund for funding to undertake a Phase 2A followed by a Phase 2B randomized clinical trial. For a Phase 2A, if we were to use clinical outcome measures, we'd need relatively large numbers of patients in each group. So instead, I chose an experimental medicine design. We recruited patients who were already scheduled for surgery, but who had large prominent nodules in the palm of the hand that were to be excised at the time of surgery. We injected those nodules with different doses of anti-TNF or an equivalent volume of placebo in a blinded manner, collected the tissue after surgery, and then analyzed it in the lab for various markers, which I'll show you in a moment. Having identified the optimal dose of the anti-TNF, we then proceeded to a Phase 2B clinical trial where we recruited patients with early stage disease, a prominent nodule with a clear history of activity within that nodule. And patients were randomized to receive either that optimal dose of anti-TNF or an equivalent volume of saline. Now, adalimumab, as you know, in patients with rheumatoid arthritis is administered every 2 weeks. However, our patients would not tolerate 2 injections every 2 weeks. So we did an end-user survey. We asked patients with both early and late-stage disease how many injections a year would they accept to control their Dupuytren's disease. Almost all said 1 injection a year. The majority said 2 and 3, and once we got to about 4, the positive responses were beginning to fall off. On that basis, we designed the trial such that patients would receive 4 injections of the anti-TNF or an equivalent volume of placebo every 3 months for a year, and we then followed them for a total of 18 months. Since Dupuytren's disease is a localized, low-grade inflammatory disease, I hypothesized that if the anti-TNF is going to be effective, it has to be delivered locally into the nodule at high concentrations. And of course, this is eminently feasible because the nodules can be identified clinically, and here you can see them on ultrasound. So that's the skin surface, there's the underlying bone, joint, and the tendon. And what we found with our ex vivo studies was that around 0.3 to 0.5 mLs remains constrained within the nodule, and after that it begins to spill out. So this is the design of the Phase 2A clinical trial. So these are patients with nodules that were going to be excised, and 2 weeks before their surgery we injected them with different doses of adalimumab. So we first injected them with 15 milligrams in 0.3 mils or an equivalent volume of saline. Next, 35 milligrams in 0.7 mils or an equivalent volume of saline. And it was at this stage I noticed that 0.7 mils was not remaining constrained within the nodule, as we've seen in our ex vivo studies, but in fact was spilling out into the subcutaneous space. Now, I had hypothesized that we would need high local concentrations of anti-TNF if it were to be effective, because this is a localized fibrotic disease. And by coincidence, the manufacturer at that point brought in a new formulation, much more concentrated, so 40 milligrams in 0.4 ml. And the great thing about this formulation was it's also free of excipients such as citrate, which should be associated with lower pain on injection. So we submitted a major protocol amendment to incorporate an additional dose cohort before we had analysed and unblinded the data. So we now had 3 different cohorts of 15, 35 and 40 milligrams in different volumes. Having collected the tissue, we then analysed it in a blinded manner 2 weeks following the injection of the anti-TNF or an equivalent volume of placebo. And what we found was that only the 40 milligrams in 0.4 mils led to a downregulation in alpha-SMA protein concentration as well as procollagen 1 protein concentration. And contrast that with 35 milligrams, so very similar amounts of adalimumab, but half the concentration, and this had no effect on either biomarker. So this study allowed us to identify the optimal dose and formulation that we could then take forward to the next stage of our study. We published the results of our Phase 2A clinical trial in eBiomedicine in 2018. Having identified the optimal dose and formulation of adalimumab to use for local administration in patients with Dupuytren's disease, we proceeded to a randomized, double-blind, placebo-controlled trial. We recruited patients with early-stage Dupuytren's disease who did not have established flexion deformities that will be treated surgically, but did have a prominent nodule with a clear recent history of progression. The first question we asked was how often to inject the nodule. Adalimumab is administered every 2 weeks for patients with inflammatory arthritis. We did not think that 2 weekly injections, which can be painful, would be tolerated. Therefore, we conducted an end-user survey. We asked patients with early and patients with late-stage disease how many injections will be acceptable every year to control the progression of Dupuytren's disease. All agreed 1, most said 2 or 3, but after 4 the acceptability started to fall off. Therefore, we selected 4 injections at 3 monthly intervals. The next question was which outcome measure to use. Correction of established flexion deformities has been used in studies for patients with late-stage disease, for example, following treatment with collagenase. However, using the converse, prevention of flexion deformities is fraught with difficulties. The available data suggest that without treatment, 20% of patients with early-stage disease progress to develop flexion deformities over 7 years, and about 35% by 18 years. These long timescales would mean that we would have to follow patients for 10 years or more, which is not feasible in the context of a clinical trial. Nodule hardness has been used to assess efficacy, for example, in patients receiving intralesional corticosteroid injections. However, in these studies, unblinded clinicians subjectively assessed, is the nodule softer? We wanted to quantitatively measure nodule hardness. Our literature search revealed that a durometer has been found to be reliable and sensitive for assessing skin hardness in patients with scleroderma, lipodermatosclerosis, and scars. Therefore, we used the same durometer. The next challenge we faced was how to calculate sample size. In a pilot study, we assessed the hardness of nodules in 25 patients with early-stage disease and the corresponding area on the palm of age, sex-matched controls. This gave us the difference and standard deviation to calculate sample size. Our pilot data also showed that we could reliably image the nodule using ultrasound scan. Therefore, we measured nodule size on ultrasound scan as a secondary outcome measure, as well as various clinical outcomes, including patient-reported measures. At this stage, I can share with you the top-line data of this Phase 2B clinical trial. We met the primary endpoint of nodule hardness and the secondary endpoint of nodule size on ultrasound scan with statistically significant differences. The full results have been submitted for publication in a peer-reviewed journal and will be disclosed on publication. In summary, I have shown you our data that the nodules of early-stage Dupuytren's disease comprise a complex ecosystem of vascular and immune cells, fibroblasts, and myofibroblasts. There is also a clear division of labor between each of these cell types. The endothelial Cells secrete growth factors to sustain the immune regulatory fibroblasts. These fibroblasts in turn recruit the immune cells. The immune cells secrete cytokines that promotes myofibroblast development. And there are distinct populations of myofibroblasts, a small cycling pool, and others that are mainly responsible for matrix production and contractility. A complex crosstalk between the stromal and immune cells maintains the chronic, low-grade, localized inflammation characteristic of early-stage Dupuytren's disease. Epigenetic regulation is also very important in the modulation of activities of the immune regulatory fibroblasts and the myofibroblasts. A detailed understanding of the mechanisms underlying Dupuytren's disease has allowed us to identify TNF as a potential therapeutic target. We identified the optimal dose and formulation of adalimumab in our Phase 2A clinical trial, and I have described the top-line data of our Phase 2B clinical trial, where we met the primary endpoint of nodule hardness and the secondary endpoint of nodule size and ultrasound scan, both with statistically significant differences. There were no related serious events in the Phase 2B trial. I would like to finish by acknowledging my laboratory team, as well as others not shown here, the clinical trials team, my colleagues in the Oxford Clinical Trials Unit, my many collaborators, Dominic Davidson and Paul Verker in particular, who recruited patients to the RID trial, as well as surgical colleagues around the country who have sent us patient samples, the patients who've donated tissue, and I'm particularly indebted to the RID trial participants who continued with all of their follow-ups, including between the various lockdowns in 2020. None of this work would have been possible without the generous support of the various funding agencies shown here, the Department of Health, and in particular Wellcome Trust for supporting the RID trial. I've received industry support for the laboratory studies and also for purchase of the drug for the RID trial. Thank you also all for listening.